Hi everyone, my name is Shantan and today I'm going to talk about an interesting topic. As the title suggests, it's the expression of PETase enzyme or insert in E. coli for plastic biodegradation. So what PETase basically is, it is an enzyme that is derived from a bacterium called Itonera sequiaces. So it is capable of hydrolyzing the PET, the plastic bottle polymer, into like organic molecules. So for our insert, we have taken the protein sequence from UniProt. So this is the ID and this is the protein sequence. Then we converted it into a DNA sequence for further analysis. So here is our expression vector, okay, and it is a blueprint of it. So we have some like cool characteristics of our expression vector. So the first one is the T7 promoter that can induce high level transcription. Then we have the ampicillin resistance gene that is useful for like cloning procedure. Then we have the multiple cloning sites. Here we have taken the NDI and XHOI for as our restriction sites. Then we have the NBL6 histac protein for protein purification, and the most important part is that it is compatible with the E. coli IBL21 strain. Here would be the construct something look like after the insertion. So in the 5' end we have the NDI. So we have this NDI, then we have the ribonucleic binding sequence for E. coli it is signed alkaline sequence, then we have the PETase enzyme. So at the 3' end initially we have the PETase enzyme or the gene, and then we have the XHOI sequence, and then we have the NVL6 stack. So after ligation, the whole construct will look like something like this. One thing to remember that the translation stops at the vector stop codon. So the whole thing kind of look like this. So for transformation and selection, we have to have ligase 8-hour insert and vector using T4 DNA ligase, and then we transform it into chemically competent DH5 alpha cells, and then we heat shock at 42 degrees Celsius for 45 seconds, and then recover it in SOC media, and then we plate it that contains ampicillin resistance gene. So we only have colonies that have that have the beta lactamase expression or the gene. So for validation of the construct, we have to do polyPCR, then restriction digest, and then Sanger sequence. All of them have one thing common, that is the 900 pp that states that we successfully inserted the PETase enzyme. And once the sequence is 100% verified, then we move on to our expression host, that is the BL21 as we have discussed earlier. So here the things become tricky and complex. So first we need to induce the cells with 0.5 millimolar IPTG. Crucially we do this at low temperature at around 16 to 20 because we want to slow down the assembly line to ensure the enzyme folds correctly rather than making something clump. So to prove that it worked, we use HTS page and Western blotting. So we will observe a Herbe band at the range of 24 to 30 kilodalton. So the Western blot also identifies our histac protein. So moving on to the purification part, we use like nickel NTA resin cans for like washing away a silver junk and get our target enzyme. So lastly, here moving on to the functional assay or the biodegradation if our experiment becomes successful or not. So for that we incubate our purified PETase enzyme with link and use HPLC to look for peaks of the small organic molecules. So if we got these peaks, we have successfully engineered a system that can turn plastic waste into biodegradable components. So this is the end. So thank you so much for listening. Thank you.